Okay, hello everybody, and thank you for joining us. If you are with us, you are with the RV FALKOR live from the Indian Ocean. My name is Deborah Smith. I'm a marine technician aboard the research vessel FALKOR, and I thank you for joining us tonight. RV FALKOR is operated by Schmidt Ocean Institute, and we are out here with a group of scientists from the Western Australia Museum. I have with me Andrew and Glenn, and they'll introduce themselves in more detail, why they're here, what they're doing. But tonight's episode of FALKOR Live is to show you what happens to a sample as we collect it in the sea and where it goes all the way to the shelf at the museum. So I'd like to ask you to ask questions, maybe hold off a little bit to the end when we have a period where we can talk specifically with the science team. They're going to tell you a little bit about what they've been doing with samples here on FALKOR with the ROV Sebastian, as well as you'll get to see some video footage of our lander and our fish trap and all sorts of information. So I'm going to turn it over to the guys in the lab, and take it away, guys. Just getting their microphones on. It's always a bit of a technical difficulty. No worries.
Hi, my name is Andrew Hosey. I am the curator of Crustacea at the museum. I look after the animals such as crabs, shrimps, lobsters, barnacles, copepods, and all the little gribbly things that live in the in between sand grains on the bottom of the sea, as well as the terrestrial groups such as your slaters or your woodlice, pillbugs, and various freshwater groups as well. My particular favorite group of animals is the barnacles. I've worked on them for the last 10 years or so as a kind of a focus. And out here, I've been managing to catch quite a few, find a few under the sea with the ROV, which has been great. Being able to actually see them live in their habitat was great. And
I'm Glenn Moore. I'm happy to be here with you. I'm the curator of fishes at the Western Australia Museum. With Andrew and the rest of the team, obviously, on a daily basis. I'm a little bit different to the rest of the aquatic zoology team because I'm the only scientist that actually works on vertebrates. Everyone else is on the invertebrates, as Andrew called them, groobly things. So I work on any of the fish in Western Australia in particular. So that includes freshwater as well as marine. And it's an absolute treat to be out here looking at the deep sea stuff because we really don't get too many opportunities to do it.
Awesome, guys. Thanks. So I'm just going to start the video, and for those of you at home, we're going to take you down under. And our first look, as I think, is sampling a fish. So Glenn, if you can tell us a little bit about what it's like to try to get the ROV pilots to sort of find a fish and what you're looking for with the video and trying to sample. Like, is it easy to catch a fish with an ROV?
It's easy for me sitting in the control room asking them to do it. Yeah. So, I mean, this is the sort of thing that we're using. It's just a standard net that you might use to catch prawns or other fish. The ROV pilots are also very skilled in the workshop, and they have modified ours so that they're much more robust, so they can use them under the water there. And you can see on the screen at the moment just how good they have become at it. I'm not sure that they've had a great deal of experience of it prior to this, and I think they've enjoyed the challenge as well. We've learned quite a bit on the way, how they can swing it. I'm always impressed how they can use the manipulator arms to catch them. I use nets like this all the time with my arm, and it's hard enough to catch the fast-moving fish. For these guys to do it remotely, it's quite amazing. You'll notice through a lot of the videos that the trick will be to turn the net over. So we want to get the fish trapped in it before we can then secure them in one of the boxes to bring them back up.
Awesome. So I think what we're seeing now is the vehicle is back on deck. How quickly do you need to get these samples on ice?
pretty quickly, actually. So most of the specimens we've been collecting so far through this trip have been living in waters of 1 to 2 degrees, and then we're bringing them up to the surface. And being where we are, we're in the tropics now, so it's probably 27 degrees at the surface in the water, and then 30 to 35 degrees in the air. And you want to get them quickly on ice, so because as they die, you want to prevent decomposition from starting as soon as possible. So putting them on ice is very important to maintain the integrity of the specimen for future work down the line.
Wow, that's cool. So Glenn, there's a picture here now. You're actually working on a fish. Why is it really important to photograph these samples? Can you talk a little bit about what you're doing with this fish?
Yeah, so this is a fantastic new record in Western Australia, and it's called a bony-eared arsefish. We've seen quite a few of them on the video, and we've managed to catch some using the nets. The photography that we're doing at this point is really critical, because this is the closest thing that we can get to do close-ups and things like that of a very fresh specimen. We have these incredible 4K cameras on the ROV, which is the first opportunity we've really ever had to get in situ photos of such high quality. To get them up on the deck, I can zoom in onto certain parts of the anatomy that I know are important for future identifications. You capture this colour that is as close to what their living colour would be as is possible, because once we've gone through the preservation process later on down the line, then a lot of that colour or the pattern is actually lost. So it's important to try and capture that as quickly as possible.
Wow, so good camera is really important then as well.
That's critical, and camera skills. And whilst we try and do as best we can, it's always nice to have somebody else looking over our shoulder and offering some advice, because we help each other out a lot, trying to make sure that we get these really nice crisp images.
Wonderful. So I've paused the video here. You are actually out on our aft deck, and it looks like you are painting this fish. What are you actually doing to the fish?
So the fish have fins, obviously, and usually when we preserve the fish, so we preserve fish in formaldehyde, which is the typical preservative medium that we use for vertebrates. And one of the problems with just preserving a fish, just dropping it into a pool of formaldehyde, is that the fins usually collapse. As you can see there on the screen now, I'm just using a paintbrush and I'm using concentrated formaldehyde. I just try and tease the fin rays out and paint them out into a sort of a natural state as much as we can. Then that will just set those fins really hard before I then put the whole fish into preservative. And that way we have the fins splayed out, because some of the characters we need to identify these things are often how many fin rays and things like that. And if they're all folded flat, it makes it very, very difficult to count.
Amazing. It's really interesting, all of the different species to see you guys bring up, and each one is taken such care. They're all slightly different. Before we move to the next section, actually, I'm going to ask, either of you can answer this. What are the different types? So you mentioned formalin, or formaldehyde, sorry. What are the different types of chemicals that you use? And is there a reason why you're using formaldehyde versus some things are fixed in ethanol, I think, and others? Can you guys talk a little bit about that sort of lab work?
Sure,
yeah, I can take on that one. So the different preservatives do slightly different things. So formaldehyde is a embalming fluid. It's very good at fixing tissues, so actually the fleshy parts, and is quite commonly used for soft-bodied animals for that reason, to keep the shape and integrity of the body of the animal, as well as like internal organs. You hear of people taking, like making microscope slides of organs and things like that. They'll use formaldehyde to make sure that the organs are actually don't collapse and maintain some structure. Ethanol we use probably more commonly in the hard-bodied animals, so such as my groups, where the soft tissues aren't necessary for the identification or the taxonomy of the group. And the reason why ethanol gets used more is because it doesn't interfere with the DNA or the molecular biology of an animal, whereas formaldehyde is very reactive with various aspects of the biochemistry within the animal. There are other alternatives out there for various different things. There was use of osmium tetroxide, which is a very toxic chemical, which is very, very good at making sure the tissues maintain their structure, and is used for high-end microscope applications, like a transmission electron microscope for really, really high detail intracellular kind of application. Technology that we will look at, the characters that we kind of look at on the outside of the animal.
So it really depends on what you want to get out of that sample later. And sort of some of it starts with what the sample is made up of, but also what you're trying to accomplish with the sample later.
Yeah, that's right. And so with fish in particular, we need to have the formaldehyde because the tissue needs to be set. But as Andrew said, the formaldehyde degrades the DNA. It breaks it down almost to a point that it can't be retrieved. So what we do is remove a little piece of flesh, a little bit of muscle, or maybe a fin clip or something, and then we put that into 100% ethanol, but we still put the whole animal into formaldehyde. If we put the whole animal into ethanol as well, they generally go really stiff, and as a morphological specimen, something to examine the features and the characters of them, that really doesn't work if they're just straight up in ethanol.
Great, thanks. Great answers, guys. I'm gonna put us back to playing some of this video. I think, Andrew, this next section is for you, and can you talk a little bit why it's important to identify these samples with these labels and what they mean, and then sort of talk about some of the creatures we'll see in some of the images after?
Certainly. So with the museum, we keep track of all of our specimens in the best possible way we can. So the labels are very important. You would have seen in that label that there was a small WAM C number. That's the Crustacean Collection Registration Number. So that is the 75,584th specimen into our database. We've got more things that aren't in the database, but that's what those registration numbers are for. And that kind of is the key piece of data that then links all the other information from that specimen. So you can lose every other piece of information, but if you've got that C number, you will be able to link it back through our digital records, our field catch sheets, to be able to fill out all the other information. So you can re-identify a specimen, however you can't re-figure out where it came from unless you've got that linked to the specimen in one way. So those numbers for us are probably the most important thing about keeping track of the animal, just having that number and having it linked through to everything else. So we keep track of where it was collected on the label as well. We give it a field identification. We never hang our hats on getting everything right in the field. There's plenty of things we certainly have not seen before in this trip. So some of it's a kind of we were making I don't want to downplay, but we don't have access to all of our normal kit that we would have in the office, so some things are likely to be a little bit off, which is best guesses that we can get for here, and we'll have to bring them back to the lab, and that's where the real long work on the microscopes, like we've got onto the side here, is where we really get the names really kind of sorted out.
Great. So I've changed the slide over, and I'm going to play it a little bit. So can you talk a little bit about this specific crustacean?
Sure. So this is an amphipod, which is a kind of crustacean that's not closely related to shrimp or crabs, but very common in shallow to very deep seas. So these guys are actually what I would consider to be giants for their kind, easily 5 centimeters long, these red ones. And they were collected in the landers as part of our baited traps that we've been deploying off the back of the FALKOR. They're attracted to baits. They spend most of their time buried in the sediment, just kind of waiting for a meal to sniff out. We haven't seen any of these animals while we've been doing our transects in the ROV. The only times we've seen them is when we did a flyby past the landers, and then obviously inside the traps on the lander. So it's been quite good having more than one method for this survey to kind of pick up more of the biodiversity that's in the canyon.
Great, and you can talk a little bit about this photo too, I guess. You're holding a lobster, right? The type we eat, right?
So this is a squat lobster. So there are fisheries in South America for squat lobsters. There's none in none in Australia. None of them quite get to the population size or big enough for anyone's quite game enough to try and catch and eat them. But that particular guy, Coelacanth rostrata, seems to be the first one we've seen on this coast. There's been other reports in other parts of Australia, as well as outside Australia in the Indian Ocean and Western Pacific. But the first time we've seen it in this part of the country, at least. And the squat lobster, this white one, this is Monodopsis. I don't know which species it's likely to be, but this group is really quite fascinating for me. The normal kind of crab that you would normally associate with being at the beach, we haven't seen very many of those. We've only really seen 2 on the ROV, and these guys not really lobsters, more closely related to hermit crabs than they are to the lobsters, so more crab-like than lobster-like, but because they've got the kind of tail, as you can see there, with some eggs carried underneath, they can tend to get called lobsters rather than crabs.
Okay, so not big enough for dinner. I get it.
Not that one, no.
They're still really cool to look at.
Yes,
so I think
definitely.
what we have on the screen here is what I would call a shrimp. Again, can you talk a little bit about this species and how maybe it's different or not different than what people at home are used to seeing?
Well, this one's a prawn, and I maintain that there is a significant difference between prawns and shrimp.
Ah, there's a good catchphrase.
will use the name interchangeably when I'm talking about food. I grew up eating shrimp because being in New Zealand, I didn't get the big prawns that they get in Australia, but there is certainly a biological difference. So this particular guy doesn't make up a fishery here in Australia, but not that species, but related species certainly are commercially caught elsewhere. It's part of deep sea scarlet prawn fisheries around the world. There is some, like I said, there are some key differences between prawns and shrimp, but we can move through this. I don't need to go through too many details. We'll be
Okay, we'll leave that for another day. Real quick, before we move on to the fish trap deployment, Glenn, do you remember at what depth that fish that you saw in the video at the beginning was collected at? I can go back and look in some footage, but I'm not...
Yeah, that one was about 2,500 metres below the surface. I mean, that particular species we've seen, I think, from around about the 2,000 mark down to about 3,500, and that's it. So when we got into the really deep dives, we didn't see it anymore.
Okay. Great, thanks. That just answers one of the questions at home. And everybody out there says hi. So hi to you guys. Hi to everybody joining us. Thanks for joining us tonight. This next section, we're going to show you a deployment of our fish trap. You're going to actually see some footage of it on the seafloor, and then we'll move on to the lander. So if there's anything unique you guys want to talk about, what really gave you the idea to sort of have this device that people at home are now seeing us sort of deploy off the aft deck?
Well,
what is a fish trap?
Yeah, this is the same fish trap that our commercial fisheries here in the northwestern Australia use. So it's a very big trap, and it's generally used in shallower waters, but given that it's never been tried before, we didn't really have any other options, so we just decided to pick one of these up and try it. It's important for us to try different methods of catching things. As Andrew said a little bit earlier, that there are lots of species that we know we're not going to be able to see on the ROV, and so we have to find alternative ways to try and sample that biodiversity in these canyons, and traps is one of the ways. So the one that's being hung over the side of the ship at the moment on that video footage is just one method, and that's obviously a very big trap that's just dedicated entirely to fish. I guess anything else could go in it. We might get some octopus or something from time to time. We haven't yet. We'd love to get one. And you'll see, I think a little bit later on, we might see the lander as well, and that's a slightly different method. I just find this technology here is remarkable. These floats, the trap is all loaded up with weights, and it gets dropped to the bottom, and these floats can handle sitting down at 4,500 metres below the surface without compressing. The technology is just fantastic, and the opportunity to use it is really quite incredible. But what we're just about to see now is really special for us to get to actually watch our equipment work. When it's remote fishing like this, you really don't know what it's doing. So to see it sitting there perfectly on the seafloor is lots of fun. This trap hasn't caught lots and lots of things, and I didn't expect it to. As I said, it's a shallow water trap, really. So designed to catch fish where the populations are usually quite dense. And if you've been following along, you will have noticed that we don't see a lot of fish down at this depth. It's quite common not to have a lot of them. But what the trap's done here is act as an aggregation device. So it's attracting fish. So now that the ROVs come down to have a look at it, we can see this beautiful face. Look at that, isn't he gorgeous? They obviously come around to explore the trap and find out what the trap is there for. It smells very strongly of all the bait we've put in it. It has a lot of electrical equipment on it so that the ship can track it and find it and know exactly where it is. And of course that's going to be letting out lots of pulses of electric current and signals and things. And I'm sure the deep sea fish, which are, because they can't see, they rely so much on their sensory systems, other sensory systems, then I'm sure they would be picking up all sorts of signals coming from this piece of metal that's just arrived in their home.
Yeah, so just to talk a little bit about that, the image that you guys see now, this piece of equipment in the center is actually a release, an acoustic release. So we track these traps and landers with a beacon, a different device that's sort of actually it's up on the actual float. So we have an ability to track the position of them as they're going through the water column, and then this device is actually what we release the weight off the bottom. So there's a bit of math that happens in terms of how much flotation you need to how much weight you need for it to sit on the bottom and not come off the bottom until we're ready to drop those weights, and then the whole device to still actually float to the surface. And so today we actually recovered the lander and the fish trap and then deployed it, and today you actually had 2 fish in it. We were in a slightly different canyon than some of the previous footage, and we were, I think, 400 meters or so, but you did get 2 fish today.
Yeah.
Yay!
That one there in the foreground, you can just see the sort of brown. Oh, there he is in the bucket now. So that's a conger eel. We've seen a few of them with the ROV, but this particular fish seems to be like to live in amongst the more structured rock. And of course, the ROV is quite limited to where it's a big square box. So if the fish has tucked itself into a little gully somewhere, the guys can fly up as close as they can and then poke the arm out. But if the fish is sort of too deep into the rocks, we can't catch them. And that's what's happened with this one so far. So to end up with it in the trap today was a real highlight for me. It's great.
So the next video is about a lander, and the lander is actually a piece of SOI, Schmidt Ocean Institute equipment that we've used on various different cruises. Sometimes it's used as an elevator for an ROV, meaning if there's extra equipment or tools or gear that the ROV needs, or if there's not enough sample space on the ROV, they may send down the lander. But on this particular expedition, you guys are using it sort of, again, like a different type of fish trap, yeah? So can you talk a little bit about what you've put onto the platform of the lander? And again, it's got a large flotation. We track it both with a beacon and has an acoustic release to release the weights so it can float back up to the surface. And this is a deployment again from the aft deck.
Yeah. So Glenn's strapped some miniature versions of this big fish trap onto there, some steel wire cages, and the brown trapezoid traps that are just a commercial cray pot that we picked up in Perth. We've meshed them in so that the escape holes are closed up, and to make sure that I capture even the small things that could usually get out of between the slats. Like I'm not after lobsters. I'm not likely to catch anything that sort of size where we're deploying it. You might also see that there's a nice white ramp on one of them. The ROV guys, and in fact all the crew, are adept at solving problems, even ones that you didn't realize could be a problem. So that's actually a mousetrap to shut the lid of one of the cray pots when the weights are released. They've hooked it up to the release so that it will just slam shut when they trigger the release for the lander itself, whereas the other one's just left open. We just kind of, the idea popped into one of their heads and we go for it, it's not going to hurt, and we left the other one open just to see if there was much of a difference, and they seem to catch the same thing. I suggest that the mousetrap actually does keep things inside better than the other one, but it's fine. And this one, when we did a flyby, we actually found that it was actually doing a much better job of catching fish than it was of catching crustaceans. 3 quite large cusk-eels, were they?
Cusk-eels, yeah.
Sitting in there quite happily, just waiting to be brought up on board. I think they regretted their decision not too long after we triggered the release there.
Yeah. So one of the things about that having that door snap shut when the weights are released, it's hard to sort of get your head around the fact that we press a button to release these weights and let that float start to bring all the equipment back to the surface. But we're still sitting here for another, well, depending on the depth, it could be 2-3 hours for this thing to hit the surface. So all of these animals have ample opportunity to escape on the way up if they really couldn't push their way out.
We could try.
So having that lid to snap shut is, I think it's a great idea. Yeah.
To have seen it go.
Yeah.
Awesome. There are a couple of questions from home. I'm going to kind of pause the feed for a second, and before we get to the next section, the sort of shelf section of our talk today. And there, sorry everybody, I'm just getting these up. There was one question from Adriana. I'm curious, how do you avoid the displacement of the fish trap by currents in its way to the bottom? It doesn't seem too heavy. So can you talk a little bit about, do you care where this fish trap lands on the seafloor? I mean, I think what...
Yeah, so for this project, probably not. It's probably not that important. This is the first time really anybody's tried to document the biodiversity here. So for us, it's almost trying to get a baseline. So it's not really that critical, the exact spot that it lands. The technicians are probably much better at giving an answer than I am, and they can predict a lot better about where the thing might land. They do all the maths and figuring out exactly how much weight we need, and of course they can overload it with weights to get a much straighter drop, so it'll drop much closer to where they've released it. If it's not that important, then they can balance the weights out a little bit, and it'll drop down. As we're talking about that, the beacon that's attached to it, they can track it. Well, it's great. When they let it go off the side of the ship, we can sit here and watch its drift. We can pick out its lat and long exact coordinates where it's going and its depth the whole way down. So it's easy enough to find, even if it's not sort of exactly where we expected it to be.
Yeah, we do have sonars on board. We have an acoustic Doppler current profiler that will give us sort of to about 300 meters or so, we can see what the current's doing at the surface. So if we expect something to sort of take off, the opportunity that we ended up with when we put the lander and the fish trap in at this particular site, the reason why we were able to film it is it was a lot flatter of an area, and they happened to only be about 100 meters off of the potential dive track for the next day. And so we actually adjusted where the ROV was going to be going, because they ended up landing almost exactly 500 meters apart, and we do a video transect every day of 500 meters. So we were able to essentially make one be the drop-in point and one be the lift-out point, and it gave us the opportunity to do that video taping. So there's another question, what do you use for weights? We have some steel plates that are stacked on top of each other, and then they're attached by a wire to the release, and those actually do stay on the seafloor. That's a small bit of debris that we do leave behind, so that is part of that science. There was one more question about, actually my niece Aslan would like to know, can an octopus get trapped in it? And she would like to know how big a sea slug can get, and what's the largest one you've brought up?
Well, I'll start with the octopus. You can have the slug. Yeah, absolutely. We could get an octopus in any of the traps that we've got. Adept at squeezing through the tiniest little gaps, and they certainly, most of them would be happy to hunt the fish that we might have already caught. So in the shallow waters, octopus are a big problem for people that are trying to catch crayfish, for example, yes, or lobsters, rock lobsters. And so the crayfish can get in there, the octopus can get in there and eat the cray that's already been caught. So if there are octopus around, we would definitely expect to end up with one in the trap, but we haven't even seen one on the ROV yet, so for some reason this place isn't really, thick with them.
No. And as for slugs, the biggest, I'm sure our chief scientist is watching now and either telling me to go bigger or smaller. We haven't picked up any on this trip. The last cruise I was on earlier this year to the south coast of Western Australia, we got a sea slug that was probably about this sort of size off the rock wall of the canyon, Bremer Canyon. Maximum size.
I'm not sure. That would be my guess. It's a bit outside our area of expertise, but
I'd like to say a foot, but I could be wrong.
Okay, there's one more question. Do you have to be careful of any poisonous specimens getting caught in the trap?
Yes, but I don't think we've come across anything here that would instantly know is poisonous or dangerous from that.
Or venomous. Yeah.
Venomous, yeah. Certainly in shallow waters, there's a lot of animals that you wouldn't need to worry about, certain fish. Octopus, little blue rings, if you had them in very shallow water, but these sorts of depths, well, we should probably be a bit more careful, maybe.
They're probably some of the urchins
yes,
often have poison on their spines. And I think we've had a few representatives of that group, but I think that the deeper water ones may not have the same amount of venom in their spines as the shallow water ones do. But certainly things like the sponges, they have tiny little spicules, so their structure is, body is made up of a real complex structure, matrix of spicules. Those little spicules you can get in your hand if you're handling them too much, and they are very difficult to get out. So they're not actually poisonous or toxic, but they actually create a lot of irritation in your skin. So yeah, we always have to wear gloves when we're handling those sort of things.
Okay, great. So we're going to quickly move on to the next portion, this sort of shelf portion. Obviously, we can't transport ourselves into the museum, but I'm going to put up a couple of pictures that the guys have kindly collected together so you can see what it sort of looks like as a behind the scenes where these specimens end up. Can you both tell me what is it like working for a museum? Like what is it that you enjoy most about working at a museum?
Well, I find our roles in the museum particularly rewarding in that we deliver a lot of knowledge to a variety of audiences. So most people think of museums as being the front of house, exhibitions, public programs, school activities, things like that, which is certainly core part of anything we ever do. Lifelong learning. So we are in Western Australia Museum. It's all ages, and we're delivering what we consider to be lifelong learning to the general public as well as tourists. So we're not just catering to tourists, we're not just catering to the locals. And then beyond that, at the back end, sections that we're doing here, some of the stuff will end up going onto display. Some of it will be part of research programs into the future, environmental management, and a whole variety of different applications down the line. Not just certainly isn't a case of just sitting on the shelf. So being able to provide information to those that need it and to the people that don't realize they need it is also a very rewarding aspect of the job.
So for me, I think one of the thrills about working at the museum is being able to do this sort of work. So fundamentally, our role is to document the state's biodiversity. And so to come out here and to try and find out what actually lives here is just fundamental to every other question about how we might protect a place like this, or whether in fact it even needs protecting at this stage. There's all sorts of things that come from just understanding what actually lives here, because we don't even know that yet. For me, the opportunity to come here is something that's uniquely in work, just to look at biodiversity. Makes museums probably, or museum research staff, which is fundamentally what we are, is that we actually have a collection associated with our research. So universities might do work on these animals, but they don't hold this collection that museums do. So that's what makes museums really, really unique. So these specimens are stored in perpetuity, so forever somebody can come and grab this thing off the shelf, make sure or confirm that the ID that we put on it or somebody else has put on it is correct, change their mind as science develops and our research changes our views on what species boundaries are. That's a permanent record that has the data that Andrew was talking about before, which is permanently attached. We know that this specimen in a jar was collected at this point on the map, at this point in time. Specimen is there, so that it's irrevertible proof that it was there. So that museums are unique in that way. No other research organisation can do that.
Okay.
So
I
what
think I'll say first is that the museum is all about exploration. From the natural history side of things, so the biology, we've got the geological history, social history, going back 1,000 years in this state, so longest continuous culture present in Western Australia, and so exploration of the state is what the museum's kind of core focus is when it comes to getting these collections as well, and we've been doing that for 120 years now.
Sorry, Andrew, can you say that number one more time, because I think you broke up. What was that number? 120 years?
130.
130 years.
130.
Okay.
Yeah.
Sorry, I just wanted to make sure it was right on the... can you guys tell us what we're looking at here? What is this, a laboratory that you actually take these specimens back to keep working on them?
So that's one of our wet labs back at base, where we'll certainly be sitting many hours on the microscope, like Mark here on the left of that photo, identifying these specimens, sorting them out to work out what they are, and where we start making start formalizing our discoveries that we're making out here.
Yeah, so here on the ship, we're basically doing the collecting, then the first pass at processing, which is all the photography, taking genetic samples for doing the molecular analyses later on, then stabilizing the specimens, so preserving them in whatever medium we need to, and then and making sure all of our data are all attached to it and complete. It's only the first step, because then we need to package it all up, get it back down to Perth safely. And then once we've got it to Perth and we can start to unpack it, everything will get looked after, entered into the databases using all of that data that we're collecting. Then we go into the laboratory to try and formalise the identifications, because as Andrew said, there's only a limit to how far we can get some of these identifications on board the ship. It's then that the hard work really begins. This is tiring, and it is challenging at times when the boat's rocking around, but this is kind of fun because we don't get to do it that often. Our work back there is when we really get serious, because that's when we start to have to put real names on things and not just a sort of a working name, if you like.
So I think this might be a young Glenn here in these shelves.
Yes.
Can you tell us a little bit about this space? And I'll keep playing some images if you guys want to talk a little bit about what these next images are showing.
Sure, yeah. This is the collection space. So all of these animals that we're collecting now will go back to the museum, and you can see it's an enormous storage area there. And once we've done these formal identifications, then everything is then put into a permanent storage, and it's usually a glass jar. Some of the bigger things need to go in larger containers. There's some fantastic examples there. And it's then stored on a shelf. We have the collection organised in a phylogenetic, vaguely evolutionary order, and we are able to go to the shelf, find out where everything is. It's well documented in the database to be able to keep track of it. But that's when I was saying before that storage in perpetuity, that collection is held on behalf of the taxpayers, the public of Western Australia, and it's available for us to work on, and it's available for colleagues around the world to work on. So some of our material will be sent around the world on loan so other people can look at it, or we'll get visiting researchers that will come into our museum and be able to sit and look at the material that we never have an opportunity to see it anywhere else. So yeah, it's a fantastic place to walk around. Lots of really cool things to look at. Anybody that gets an opportunity to walk through the collection, which of course is not open to the public, people just like to walk through it, and it's incredible. It's a really interesting place. Even after many years of working in the museum, I still love walking through there and just admiring.
Well, maybe
Given it being a bit of a scientific library in some respects, in that people take specimens out on loan, we'll get specimens in on loan, and work through our research programs as we need them.
Well, maybe at some point we'll get you guys to give us a virtual tour of that. So.
Yeah.
We could do a ship to shore
Yeah,
Short of ship.
inside the museum, behind the scenes. Awesome. So we've got a few questions coming in from home, and I ask everybody now at home, again, thank you for joining us. If you have questions for Andrew or Glenn, please write them in now, and I'll relay them to them. We have one from somebody here. There's actually two questions. So they say, awesome work. I have two questions. Are you looking for specific species or groups? And do you also catalog the bycatch? So maybe you guys can talk a little bit about the difference between what we're doing now and bycatch, and then and specific species. And this is a great segue to also talk about the taxonomic tree.
Well, I'll start with are we looking for certain things? And for the most part, I would say the answer is no, because this project is specifically aimed to just document the biodiversity of this deep sea canyon that we effectively know next to nothing about in terms of the biology of it. So we're basically looking for anything. We just need to know what's here. And so with the ROV, the way that it's collected, it's highly selective. So we basically have no bycatch using the ROV as a sampling method. Occasionally, when something might be picked up, when it comes on board, we'll find there's other things on it. There's little parasites or commensal animals, so animals that are using another animal to live on. some people might call it bycatch. Certainly our scientific team call it another fantastic specimen. So there's effectively no bycatch in using this method of biodiversity assessment.
Yeah, we certainly have our own favourites, and we're certainly keeping an eye out for them. So Glenn obviously is watching every fish like a hawk. I'm keeping an eye out for any crustacean that happens to be crawling on the seafloor, but we've picked up some really random animals that nobody on board has direct experience working on. You may have seen on some of the other posts the big giant hydrozoan, which is a massive polyp that looks a lot big, like a bit like a flower. So none of us on board has any kind of research experience or expertise in that group, but it's such a unique sighting of an animal that we thought, well, we need to find out what that is. So we picked it up, catalogued it, take it back, and we'll find experts somewhere in the world that have experience, and we'll get them to examine and confirm what it is.
Awesome. So, okay, again, another call out to those of you at home. Andrew, would you mind talking a little bit about the taxonomy tree and why and what is important about each of the different levels? So, what kind of, for the people at home who aren't familiar with identifying species, and for example, what is a species versus a family versus I'm going to get it all wrong because I'm not a biologist, but if you could talk a little bit about what that means.
Sure. So to make order out of chaos, we order things into boxes, for want of a better word. We're sorting these animals into groups, and so about 250 years ago, Linnaeus came up with the current taxonomic naming system that we still use. It's changed a few times over the years, but it's mostly the same still. So at the top end, so the broadest category is what's called the phylum. And so things like arthropoda, which has your insects, spiders, crustaceans. You've got chordata, which has got things like your vertebrates, groups, mammals, reptiles, fish, mollusca, octopus, things like that. So that's with the top end, and then it slowly gets more and more specific to individual species at the bottom. So you've got various levels, and these are, in a lot of ways, these are a convenience of a way of making sense. It's arbitrary. We've gone, well, these things look quite similar, so we're putting them together. So our methods for developing these have gotten more sophisticated over the years with different analyses, mathematical modeling of what we call character states and things like that, where we're actually kind of trying to quantify what some of these characters that these animals have, how they're actually important for the evolution of the group. So we try to have these boxes that we're putting together as what we consider to be natural, so evolutionary sound, so that all of the animals inside that box are actually more closely related to each other than they are to something in the next box. It's quite complicated, and there's a lot of protocol and procedures around there, especially when it comes to naming and things like that. And as genetic data is coming in, we're starting to integrate that into the system a lot more closely now. Making for interesting times for people that didn't come through school using it, knowing the genetics as closely as I did. So I'm still a bit of a novice with genetic data, so I'm myself up there. But yeah, that's kind of where it works. So I probably skipped over a few things.
Well, I'll just say that the species is fundamentally what most people want to know. So when you see something, people say, what is it? So that's the lowest 100%. Level or the highest level of identification that most people want. And so when we're trying to decide whether two species are different or the same, and whether they're related into the same genus, which is the next level, I guess in some ways it's rather arbitrary, because as humans we like to put things in boxes. That's how we make sense of the world. If we have to put these things into boxes, then part of that is we hope that those boxes reflect evolutionary pathways. It doesn't always, but that's what we try and do. And most of taxonomy's history, it's been based on morphology and the assumption that things that have common features probably come from a similar evolutionary pathway. There's lots of differences in that, because things can evolve the same character in different pathways, but if you cover enough different characters, you'll get them to a point where you think it's right. Adding genetics to that, more and more we now understand genetics and these really complex mathematical probabilities that we can apply to it, can start to test whether some of our morphological trees that we hope reflect evolution are also reflected in the genetics, which is really a real sharp look at evolution. It's probably the sharpest, clearest view of evolution that we can get. Yeah, marrying the two up is when they match, it's great. If they don't, then there's a lot more questions to answer.
Yeah.
Great, thank you so much. So we have one question from home, and I'm actually putting it up on the screen in front of the guys because I don't know how to pronounce it. Can you read the question and then answer it?
The question is, are there any chances of finding any coelacanth in any of the canyons? That would be pretty amazing. My answer would be probably not at the depths that we're working. So the coelacanths that have been collected in South Africa and in Indonesia are in much shallower water. I think, off the top of my head, because we don't have it in Australia, I'm not entirely sure. I think they're around about 300 metres is about the deepest one. So I think the shallowest dive we're doing is at around about 2,000 metres, or just over 2,000 metres. So I think we've surpassed what we would expect to see, the depth that we'd expect a coelacanth to live. But if we found one, I reckon it'd be pretty big news.
Okay, I don't know what they are. Is there like a generic name for those of at home?
The coelacanth is its common name.
Okay.
Latimeria is its generic name, which probably doesn't
help
anymore.
me. Okay, well, Google it. This is everybody's tonight's Google homework. I gave homework last time. This is your new Google homework.
Yeah, they're very interesting fish. Unfortunately, they're not on the Australian list yet. We never had one recorded in Australia. Yeah, I would recommend, if you want a bit of homework, then reading about coelacanth. They're fabulous.
I just got from Jimbo, they're basically living fossils. Okay.
100%.
So,
yeah.
Awesome. One question from home, from me. My dad's asking, will samples go to any other countries, typically?
Yes. So in the first half of this trip, we had Greg Rouse from Scripps Institute in San Diego. He was on board. He's seems to be expert in everything. He knew quite a lot of what we were seeing underwater before anyone else had a clue what it could be. And he's getting a lot of our worm samples sent to him directly as a result of this.
On loan.
On loan.
Yeah.
None of the specimens are going to be retained by any other country at this stage. As part of the species description and taxonomic research phase, there will be designated specimens as type specimens. So these are the key specimens that are a part of the description of the species, and some of them will get spread to other institutions as for safekeeping rather than having all our eggs in one basket. And I'll just explain the concept of the type specimen. So the holotype is the standard for a named species. If you propose a new species, you designate a holotype, which is then what holds that name. And so any other specimen that is collected that you identify as that species,
that's a good one.
is by default compared to this one holotype. So what I like to compare it to is that in France, there is the original metric measures. So the kilogram, gram, meter, they have the, I don't think they call them types, but it's the measure. So every other meter or gram that's ever weighed everywhere else in the world is by default compared back to that actual thing that's made out of platinum, I believe, for them, and they're kept in a certain vault, and every country will have models of them around the world from which they calibrate everything, theoretically at least. So yeah, it's a standard. It's the standard, and if there's ever a dispute, the holotype is the key specimen that will solve it, settle it. No, in some cases not.
Yeah, but that's part of the process of describing a new species. I'm sure there will be new species out of what we've found here.
100%.
The process of describing a new species can take many years, and lots of collaborations in many cases. As Andrew said, the holotype is that one specimen that holds the name, as the name-bearing type, can never be replaced. So if that specimen is destroyed for some reason, if there's a fire or whatever, you don't replace it. It's a one-off. What we do is collect or describe, when we describe the species, we have a whole range of other specimens that are designated as paratypes. They reflect that that species, but they sort of might cover some of the variation you would see in shapes, or some of the variation in colour, or fin ray counts, or
Males,
number of
females.
spines, males, females, juveniles, adults. They would for a broader range, because of course just think about humans. You don't have a holotype for humans, but which human would you choose to represent humanity? I mean, there's not one individual. Sorry, I just got distracted because J. Rod thinks he should be the representative.
We've got the ethanol. Put it in.
So the holotype is close as you could get to the sort of best individual, but of course there's so much variation, and one individual isn't going to capture that variation, so you have to have a bigger range. Well, ideally you do.
Okay, and just for you guys at home, somebody asked the question, where are the Ningaloo canyons? And so we just put up the map. I zoomed out. We are in the sort of western side of Australia here. And we can zoom in, but we've been mapping in the evenings and diving with the ROV in the daytime. And you can catch all of our ROV dives live on YouTube and Facebook.
So this is the Indian Ocean we're in?
Indian Ocean, yeah.
There you go, right up in the northwest corner of Australia.
So this is the Cape Range Canyon, which we did a lot of the first half of this cruise's dives in, and we've just now started to work on this southern canyon. The mapping is actually done. I've yet to import it into Google Earth, but you can see here tomorrow we're actually going to be diving at this CL15 site. So join us tomorrow, hopefully. Any more questions from home? Now's your chance to ask Glenn and Andrew any new questions. So Maureen asked, who gets to name the new finds?
Us?
Yeah, hopefully. So I guess ultimately it would be nice if we could. The reality is, for many of the things, we're not the experts, and there are already existing experts around the world. We have expertise in the things that we know well, but there's lots of other things. We just have to find collaborators or offer it to them to examine. Of course, it is all retained at the West Australian Museum, but it does get loaned out. So it's usually the expert in the field, and they've developed their expertise over many, many years, decades in many instances in taxonomy. And so familiar with all of the characters and all of their species, and so for them it's much faster for them to interpret the new thing, be able to fit it into the phylogeny, the species boxes, as we talked about.
Yeah, we're a community of scientists around the world that focus on the species description and discovery, and because there's so many to do, we tend to kind of into our little specialties and work our way through it. So with the lander, we've picked up a lot of those red amphipods. I can't say that I'm particularly skilled with that group of animals. So I've got a colleague, Rachel King, based at the South Australian Museum, who's kind of keen to have a look at some of those animals that we've picked up. And from there, I hope to work with her to describe and document the fauna that we've picked up.
Hopefully we're involved in most of them, but probably not all of them.
So we have one more question from home. And again, you guys, you have about 1 more minute to ask your questions, so please write them in. These guys have to get up very early in the morning, and I've kept them up way past their bedtime so they could join you here. What has surprised you the most on this expedition?
Good question.
Yeah.
Good question.
Surprising. I've done a couple of trips with these guys over the years on the FALKOR. Once before, they had a dedicated ROV of their own. And one of the things that I've found quite surprising is how quick they are, crew across the boat, how quick they are to actually adapt to situations. So the ROV team learning how to catch fish with a net underwater as if they were just scooping it from a pond. It has been amazing. Underwater, contrast with other parts of of Western Australia. So it's interesting seeing the different seascapes. So I'm not a geologist, so when I see interesting geology underwater, I don't really understand some of it, but it always stands out. So one of the weird things that we saw on dives ago maybe were some cliff faces, which were probably 50 meters high, which had black and white layers. You just see it. It was like a mime's top. So if I wanted to describe it any other way, I can't. And the followers that you got, the FALKOR has over the YouTube and Facebook streamings, you get people that absolutely know what they're looking at along. That surprised me a lot. We had a geologist, Karen, knew exactly what those were. They called them turbidite, and it was just different layers of sediments just coming down. One layer was should have been a marine sediment, and then the next layer was more of a terrestrial one. So the idea that there had been either river or rainfall that had washed out a whole lot of sediments in sort of seasons of, well, whether it's actual seasons or some other process, we weren't too sure. But yeah, just layers. That surprised me a lot.
Yeah, I think for me the thing that surprised me is just how much sedimentation there is. It's obviously, I guess, the deep canyon, when you think about it, it's all the sediment that's in the water that's falling down is getting caught there. We have the tiniest little fragile animals that attach to rocks or just hanging there in the water, and they can survive in this constant stream. There's a snowstorm of sediment that keeps raining down on them, and they manage to flick it off their tentacles or brush it aside and survive without being smothered. It's amazing. It's really amazing.
Okay, so we have one more very specific question from home. Have you collected samples for and fauna organisms specifically below 2 millimeter sizes?
We haven't.
Not a lot. We haven't targeted that sort of group of animals. We have taken up sediments and sieved them. So you would have seen us doing our little push it moments with the push core, hearing us probably sing over the live feed. So I think I'm cutting out a few times. We will take those sediments up and we will then sieve them through a 500 micron mesh, as well as using the suction sampler or slurp gun on the ROV to kind of just vacuum up a fair bit of sediment as we're trying to collect something else that's in amongst it. However, we haven't done a lot of collecting to try and target those sorts of groups.
Yeah, there's, as you said, a bit of suctioning that's come up. So our two mollusk experts on board are very keen on the micromollusks. So there's been quite a bit of excitement in the lab here when they're going through sediment that's actually just come up by collecting other things.
Yeah.
And they've managed to find all sorts of fabulous things. We've been on field trips with them where they just spend all day, all night sifting through mud and looking for it. I think the opportunity to do that here is a bit more occasional. They're not taking huge samples. I think it's probably a bit busy.
So many other things to collect.
That's right. Yeah.
Awesome. Well, I'm going to say that I think our time is up. We have been on air for an hour now. So if you guys have any final words or comments, if you want to make them, I want to thank everybody at home for joining us.
Yeah, absolutely. I think it's fabulous that we can talk to you sitting there at home and we're way over here, and not only the other side of the world, but it's floating around out in the middle of the ocean, that we can have this sort of opportunity. It's great. Make sure you tune in for the rest of our dives.
Yes.
So this has been a great experience being out on the FALKOR. This is a great setup. There's a good crew, good food, good times. Taking us to places that we would never dream of being able to get to any other way. So, fantastic opportunity for all of us on board, and we're hoping to get them back across to Western Australia again in the next couple of years.
Great. Thank you, Glenn, and thank you, Andrew, for joining me tonight. Thank you, everybody at home, for joining us for all your questions. If you have any more questions, feel free to reach out to us on Facebook. Again, like we said, you can learn more about this expedition at schmidtocean.org. You can follow the expedition. There are lots of blogs on different topics there. You can also watch our live ROV dives from ROV Sebastian on YouTube and Facebook any day that we are in the water. We are planning to do an ROV dive tomorrow, so hopefully we will start that first thing in the morning. So again, thank you for joining us. Have a good evening or day or afternoon or morning, wherever you are in the world. Stay safe. Wash your hands. There's a very important message from ROV Sebastian. If you haven't seen that video, watch it. Wash your hands. Stay inside. Stay safe. And thank you to everybody at home. We miss you and love you.